The overall goal of this procedure is to observe the development of original and metastatic tumor formation in epithelial ovarian carcinoma.
This method can help answer key questions in the gynecologic oncology field, such as understanding the tumor biology of peritoneal dissemination. The main advantage of this technique is that an orthopatic inoculation of human epithelial ovarian carcinoma, or EOC cells, is performed through a retroperitoneal approach from the dorsal flank.
To prepare a human ovarian clear cell carcinoma cell line, plate and maintain ES2 cells in rPMI 1640 medium with 10% FBS and Penstrep. At 37 degrees Celsius and 5% CO2. In the logarithmic growth phase, remove the old medium from the culture and use 3 to 4 milliliters of PBS to wash the cells once. Next, add 1 milliliter of trypsin EDTA and rock the vessel to cover the entire monolayer. Remove and discard the trypsin solution and return the cells to the incubator for 3 to 5 minutes. After examining the cells under a microscope to ensure the cells have detached and are floating, add 5 milliliters of fresh serum-containing growth medium to inactivate the trypsin and resuspend the cells before transferring them to a 15 milliliter conical tube. Centrifuge the suspension at 300 times G and room temperature, or 4 degrees Celsius, for 5 minutes. Then, carefully remove the supernatant and use 5 milliliters of growth medium to resuspend the cells. With trypan blue exclusion, count the cells. And use growth medium to resuspend them at a density of 2 times 10 to the 8th cells per milliliter. Then, place the cell suspension on ice. Thaw frozen extracellular matrix or ECM-based hydrogel aliquots in an ice bath. Add the cell suspension to the hydrogel at a 1:1 ratio and thoroughly mix. Then, place the cell suspensions on an ice bath until use. To carry out orthotopic inoculation, after anesthetizing an 8-week-old female nude mouse according to the text protocol, perform a toe pinch to verify depth of anesthesia. Place the anesthetized mouse on an operating stage belly side down. Then, disinfect the surgical area with several washes using a chlorhexidine-based scrub and alcohol. Next, make an approximately 2 cm vertical incision near the spine between the right costal arch and the femur. Place a sterile surgical drape around the incision site. Then, fix the incised skin surfaces with clamps to maintain the opening. Now, make a second 1 cm incision at the parietal peritoneum under the first incision to open the abdominal cavity. Then, clamp the incised abdominal surfaces. With a clamp, clip the fat tissue that surrounds the ipsilateral ovary and fallopian tube to remove the ovary from the abdominal cavity. Then, carefully place the ovary and fallopian tube on gauze before placing the mouse, belly side down, under the dissecting microscope. Transfer the cell suspension to an insulin syringe. Then, immediately and carefully inject 1 to 2 microliters of the prepared cell suspension into the ovary. For several seconds, keep the needle in the ovary to ensure that the hydrogel sets. After gelation, the injected tumor cells will remain in the ovary. Carefully, return the ovary into the body. With sterile medical silk, suture the second incision before using wound clips to seal the skin. Following the procedure, place the mouse in a separate cage on clean paper with a hot pack to keep the animal warm until it has regained sufficient consciousness to maintain sternal recumbency. Return the animal to its cage for rearing and monitor the mouse for 3 to 7 days following surgery. For signs of pain or infection, administer analgesics or infective agents according to the text protocol. 2 to 3 weeks after inoculation, sacrifice the mouse, collect the samples, and carry out analysis according to the text protocol. As seen in this figure, after 2 weeks, 5 of the 6 mice inoculated with the clear cell carcinoma cell line ES2 had a tumor in the inoculated ovary, while the contralateral control ovaries were tumor-free. Moreover, 2 of the 5 mice showed multiple peritoneal disseminations with ascites and cells metastasized to the liver. H&E staining showed a large number of ovarian cancer cells in the inoculated ovary, and the cell morphology was the same as that of the parental tumor. In contrast, no cancer cells were observed in the contralateral side.
Once mastered, this technique can be done in 30 minutes if it is performed properly. While attempting this procedure, it's important to remember to place the cell suspensions on an ice bath until immediately before use. Following this procedure, other methods like using an in vivo imaging systems can be performed in order to answer additional questions like the dynamics of the spread of cancer cells from the primary tumor. After its development, this technique paved the way for researchers in the field of oncology to explore tumor metastasis in gynecology. After watching this video, you should have a good understanding of how to inoculate human EOC cells at an orthopathic site using a retropenetrate approach from the dorsal flank. Don't forget that working with the cell lines derived from a tumor can be extremely hazardous, and precautions such as wearing gloves should always be taken while performing this procedure.